What is biotechnology? Okay, so what can you do with the biotechnology? You can create a glowing rat. You can create a plant that can produce different kinds of fruits. You can make a crop that can resist the pests. So what we do here, we will insert certain genes to the DNA of the organism so that the organism can get desirable outcome. First, suppose here you take the glowing rat. Normally, it's a normal rat, but you modify the genes of this rat. Now it's having bioluminescent genes. So I'm modifying this rat with a bioluminescent genes so that now the rat will produce certain proteins that can glow. The same way here, I can make a salad tree which can produce multiple type of fruits. Even I can engineer the crop so that it can be pest resistant, so that can produce natural insecticides. Before the crop couldn't produce, but after modifying that, now the crop is able to produce natural insecticides that can kill the pests on its own without using any external insecticides or artificial insecticides. Is that clear?
Yes, sir. Yes, sir.
Okay. So how does the biotechnology has started? Okay. So first, the biotechnology we use in everyday life. For suppose the production of these different products like curd, bread, wine, beer, cheese. Okay. So all these we technology principles. So how does it started? Let's see. So initially it was started 10,000 years ago. Okay. So by a mechanism called selective breeding. So what does that mean? We will select two different types of organisms within the same species. For suppose here there is a hen which is very skinny, but it can produce number of eggs. And there is another hen which is hefty, but produce very less number of eggs. Then I do the crossbreeding. So I will get a hen that is hefty and can also lay more number of eggs. So I'll be getting both the desirable characters from both the hen. So what is the desirable character here? Laying more number of eggs. And here FT. So I'll be getting the desirable genes, both the desirable genes within this organism. So
But
which
there's
will
still
start,
chances.
yeah,
So there's still chances that there's also possibility that there'll be a chicken form which is skinny and lays few eggs.
yeah, that can be possibility. But I'm modifying here the genome. I'll tell you how we'll modify that genome in the
Mm.
next. Okay. So right now you just remember we'll be taking the desirable characters from both the N and we are making the new N that is having both the characteristics. Okay. So that we call it as selective breeding. So with the selective breeding, you can improve the quality of the outcomes, quality of the yield. Even you can make the crops that are disease resistance. Okay, so you can make disease resistance crops through this process. So remember, it was started 10,000 years ago. It's not now, very back. Okay, and if you see 9,000 years ago, it started in the Egypt, the production of the bread. Actually, these Egypt guys, they have a tradition, they have a norm that is offering food, or they have a practice, like it's a custom that they will give or offer to the dead ones. You know, the Egypt mummies. Okay. So they will offer to these dead ones the food, all these things. In that, the main thing they offer is the bread. And how does they make a bread? By using the flour, sugar, olive oil, water, yeast. So there is a timeline that in the 9,000 year back, they started making the bread production, and here we are using the yeast that will produce the bread, so which also comes under the biotechnological process. And if you come 4,500 years, this is also happened in the Egypt. So there is a wine production. So 4,500 years ago, they used the grapes for making the wine. Okay, so to in order to prepare the wine, they had the yeast. So remember, to make the bread or to make the wine, we need yeast. Okay, and they store it for longer time. So how old? The fermentation happens much more, and that gives the very strong wine. So that's why the older the wine, it costs so much. It is very costlier the older it is. And does anyone know who is Edward Jenner?
The one who did the chicken pox vaccination, like the father of vaccination, something like that.
Yeah, yeah. So he's the person, he's the first person who has discovered the vaccine. Okay. So actually, there is a disease called the smallpox. So actually, there's a village that is affected with the smallpox. In that, he discovered a vaccine that is a dead organism, and he is inoculated that organism to a small guy. I think 8 years old boy. So what happened? The guy was affected. This boy was affected with a mild fever, but then recovered from the disease. And when the smallpox was got to this person, he was still able to survive through that disease. Okay, and this is the very great discovery. So now it is the greatest discovery within the medicine itself. Okay, so we after this discovery, there's a lot of contribution for the vaccine production, which has saved lakhs, millions of lives. So, in what diseases do we use vaccine? So, tell me some examples. We use vaccine for polio,
So COVID.
recently COVID, tuberculosis,
All the viruses, right?
rabies. Not all the viruses. So, suppose we don't have a vaccine for HIV, it is continuously mutating, and we cannot produce a vaccine for HIV. So,
Mm.
what is vaccine? Vaccine is nothing but that organism, but it was killed. It killed.
Yeah.
Okay. So, vaccines can be prepared two ways. That means either you will directly take the virus and you kill that virus, or you will be taking the antigen from that virus. We will give inoculate this liquid into the person's body. So based on that, when the virus enters into our body detects it's a foreign material. Our immunoglobulins, they will find out that these are the foreign materials and they will call all the white blood cells. Okay. In that memory cells also, they will come and they will scan this virus and they keep the memory. So whenever this virus comes to our body again, now our body know that who is this guy, who is this foreign guy, and it's now ready to take the action. But what happens if the disease comes to the body? So that means our body doesn't know what virus or who is this foreign material. So we has to study about this organism, keep the memory cells, start making the antibodies against this antigen. By that time it do all these things, our body is almost affected with the virus or bacteria. So we should be ready with that. So in order to that, we are giving the vaccines. So now our body will know who is our enemy. So it is ready. It is already prepared antibodies against this virus. So whenever the virus comes to our body, now it is ready for releasing the antibodies. Understood? So what about Alexander Fleming? Who is Alexander Fleming? Any idea? The person who accidentally discovered the antibiotics.
Is it penicillin?
Yeah.
That is vaccine, right? Like,
No, antibiotics. Okay. So what happened is this guy was testing some bacteria. Okay, and he left those cultured dishes without washing it.
So is it
So
penicillin?
yes, it is a penicillin.
Yeah.
Okay, so actually he's performing the experiment in the petri dish, and without cleaning it, he left that. So when he saw the next time, there is a mold. So mold is a fungus. Okay, so there is a mold growing in that petri dish, and wherever this mold is present in that area, there is no bacteria growth. But other than this fungal area, the bacteria growth is seen. Upon testing that, he understood that these fungus were releasing certain secretions. Those secretions were killing the bacteria. And he named them as a penicillin. So that's the first antibiotic that was discovered. Okay, so what are antibiotics? These are the medications which kill the bacteria. So they are very helpful for fighting against the bacterial infections. So see, he extracted the antibacterial toxin from the mold and named it as a penicillin. And that was the first antibiotic that is actually accidentally discovered. Understood. It was happened in the year 1928. And who is the scientist who discovered the antibiotics?
Alexander Fleming.
Alexander Fleming. Okay. So next one, in the 1950, it's the beginning for the modern biotechnology. So that's the era, that's the new era where the modern biotechnology has started. So before that, okay, so remember 1950, it's the new era where modern biotechnology has started. Till then, it was all the ancient biotechnology methods. So what happened after this? First, suppose if you take 1953, we got to know about the DNA structure. So we understood about the DNA structure. Okay. And we understood that most of the organisms, the genetic material is DNA. Okay. And the scientists were thinking, what if we change this genetic material? So we know that DNA is the genetic material that's storing all the genes, and it is the main thing that is producing the proteins. So if we somehow modify this DNA, can we also modify the proteins? This is what the scientists were questioning at that time. So scientists were thinking, why don't we modify this DNA so that we will get the desired proteins? Understood? So from that, we got to know there's a modern biotechnology. It's the technique that allows us to introduce a set of desirable genes without introducing undesirable genes into the organism to get the desirable products. For suppose we know there is an organism, it is having particular genes, and these genes are very desirable genes. So what we do, we take these particular genes or whatever the DNA that is coding for these particular genes, and we will insert into another organism. Now this organism is modified, so it also start producing those particular genes. One of the example is insulin. So what we did, first initially we used to use the insulin from the uh, pig. Okay, but if you see, even though it's a producing insulin, but that insulin is somewhat different from our insulin, whatever our pancreas are producing. Maybe it's 90% same, but still that 10% it's a different. So that leads to our body to reject. So whenever we are giving the insulin from the pig, our body shows so much of side effects, adverse reactions. So what the scientist has done after this modern biotechnology, they started extracting the insulin genes from beta cells of islets of pancreas, and they introduced that into the E. coli or into some other bacteria. So now these E. coli got those genes and they start producing this insulin. And if you see, whatever the insulin that is produced, it comes from the same DNA that we have. So the insulin is also same. So our body feels like it's our insulin, it's our material, it's not a foreign particle. Our body start accepting and showing very least side effects. Understood? So what do you mean by introducing the desirable genes without undesirable genes?
So could you repeat the slide? I didn't understand anything.
Okay, so let me tell you this. First, suppose before we used to extract insulin from the pig. So why do we need insulin? First, suppose let me tell you why do we need insulin. Okay, you are taking the food. That food digests in the stomach, in the small intestine, and it will be absorbed into the blood. And whatever the glucose that is present in the blood, it has to absorb into the cells. In order to absorb into the cells, we need insulin. Suppose think like insulin is an entrance ticket. So what does actually the insulin do is, there is a receptors present on the cell membrane. This insulin has to bind to those receptors. When it bind to those receptors, there are some internal activities will happen that trigger and open the gates that can pass the glucose. So here insulin acts like a ticket. That ticket will allow the opening of the gates. So when the gates are open, the glucose can go inside the cell. Understood? So now what happened is the beta cells got damaged, or the beta cells of pancreas were not working very efficiently. When it is not working very efficiently, the insulin production got reduced. When I don't have enough amount of insulin, my glucose was just circulating in the blood and it is not absorbing into the cells. So that result in increase in the glucose levels of the blood. So we call it as hyperglycemia. So what can we do? Our pancreas cells are not working well. So we cannot produce the insulin by ourselves. We need to depend on the others. So initially, when there is no discovery of the human insulin, we used to extract the insulin from the pig and we are giving the patient. That is working, but the patient is showing so much of adverse reaction because still it's a pig's insulin. So our body is not accepting it. It is rejecting it. It's showing so much of adverse reactions whenever the pig insulin is given. So what scientist has done is they extracted the genes from the beta cells of the human and they inserted these genes into the E. coli DNA. So whenever E. coli makes the protein, it also makes the protein of insulin. And we filter this insulin and we give it to the humans. As it is made from our own DNA, so our body accepts it without any rejections, without showing any adverse reactions. Understood? Is that clear?
Yes, sir.
Sir.
Srikar?
Yes, sir.
Anyways, we'll discuss about this in detail. There is a complete detail applications of biotechnology. We talk about this insulin. Okay. So just remember what is modern biotechnology. That is nothing but manipulation of the genetic material. That means you are changing the DNA so that the end proteins were also changed. And that you are doing so that you are getting the desirable proteins. Understood? So remember, the modern biotechnology technique is the introducing the set of desirable genes into organism so that the organism is modified and it gives the desirable products. You are First, suppose you are taking the desirable DNA from an organism and you are introducing this DNA into another organism so that this organism now able to produce the desirable proteins. So that is called as the modern biotechnology. Understood? Is that clear for everyone?
Yes, sir.
Yes, sir.
What about the others?
Yes, sir.
Sir, Gayana,
Yes, sir.
Gayana, I'm very happy that you joined today's class.
You're welcome, sir.
Okay. So modern biotechnology were classified into two principles. The first one is genetical engineering, and the second one is bioprocess engineering. So let's see what is that genetic engineering. Okay, so genetic engineering is nothing but alter the genetic material. That means you alter the DNA of the organism so that the genotype and phenotype is also changed. That means you change the DNA of organism and whatever the end products, the outcome is also changed. It can be the genotype or phenotype. That means you will change the DNA. Suddenly the organism will start growing very tall. You have changed the DNA. Now the organism become pest resistance. You have changed the DNA. Now it start giving abundant of product, abundant of fruits. So the altering of the DNA or the altering of the genetic material so that the outcome is also changed and it gives the desirable outcome, desirable proteins. So such type of organisms which are changed with the DNA, we call them as genetically modified organisms. So everyone write down this definition. So what is meaning of genetic engineering? Write down this definition. It's the technique that alters the genetic material of the organism, which will change the genotype and phenotype of the organism. Please write down this definition. So let's see how do we do. So tell me how many of you remember the process translation?
Uh,
Or how many of you remember the central dogma? What is central dogma?
Sir, it's the transfer of DNA, right? From DNA to RNA to proteins.
unidirectional flow of genetic.
Okay. The flow of genetic information, we call it as central dogma. For suppose from the DNA, you are making the RNA, and from the RNA, you are making the proteins.
It's from DNA to DNA, then RNA.
DNA to DNA, DNA to RNA, RNA to proteins. So what do we call the DNA to DNA? So what is DNA to DNA is called as? Replication. So tell me replication, which type of replication does it happen? There's a replication. We specifically call it as semi-conservative replication. So what is meaning of semi-conservative replication?
Each of the daughter strand have 50% of the parent strand.
And 50% is newly sensitized. Okay.
Yeah.
So, and DNA to RNA, what is that process called?
Transcription.
Transcription. And RNA to proteins, we call it as
Translation.
translation. So if you see here, why we call this as a transcription and translation? What is the word meaning transcript, translate?
You're using different you're going in transcription, you're using the same you're using nucleotides, but in translation you're going from nucleotides to amino acids, so you're using different
Okay.
monomers.
Yes. So if you see the DNA, it's made up of nucleotides, and RNA, it is also nucleotides. That means the language is same here. Both are nucleotides, but the format is different. For suppose DNA is a double-stranded, RNA is a single-stranded. So the format is changed, but the language is same. That's why we call it as transcript. Transcript means the same language, but different formats. We call it as transcript. But if you see RNA and proteins, it is nucleotides, and proteins are made up of amino acids. So this is different language and this is different language. So as you're changing from one language to other language, we call it as translation. As you're translating information from nucleotides to amino acids. Okay. So if I change something in my DNA, my RNA will be changed and my proteins will be changed. How do I change my DNA? Either I'll be introducing the new nucleotides into my DNA sequence so that my mRNA will also have the new nucleotide sequence so that it will result in making the different protein. That process is called genetic engineering. For suppose you see here, So we know organisms, they have a gene that will code for certain amino acid sequence. So that gene we will say in terms of codons. Every three codons will make a particular amino acid. So initially this is the codons, a series of codons that is making a series of amino acids. But then I added extra amount of gene so that there is an extra amino acid that was created. That will result in change in my protein. Understood? So see, genetic engineering that will allow the introduction of the new nucleotides in an organism so that the genotype of the organism is also changed. That means as I'm introducing the blue color here, the blue color, the new DNA into the my DNA, so that instead of having only up to the blue color of amino acids, I got extra additional one color. So as the protein here changed, we call it as genetically modified protein. Or these organisms, we call it as genetically modified organisms. Okay, which will result in different amino acid sequence, thereby change the phenotype of the organism. So for suppose, what is meaning of phenotype? Okay, you don't know what is phenotype, genotype. I'll tell you, phenotype is nothing but the physical outer appearance of a particular character. Physical outer appearance of a particular character. I think you might have studied in 10th. So for suppose you
Yes.
take the height of the plant, you take the shape of the seed or color of the seed. Okay, these are all physical outer appearance. So when I change my genotype, the phenotype will be affected. For suppose instead of having round color, now the seeds are having, sorry, instead of having round shape, the seeds are having now wrinkle shapes because I changed the genotype. Instead of having just a normal height, it will be having additional height. Instead of having a sour taste, now the fruit will be sweet in taste because I modified the gene of the organism. So the phenotype will also be changed. Understood?
Yes, sir.
Yes, sir.
So that's what, in rough shell, what is genetic engineering. So the next one is bioprocess engineering. We'll be studying genetic engineering and bioprocess engineering in detail, step to step. Just these are basic definitions. So what is bioprocess engineering? Okay, so genetic engineering where you are modifying the organism. Okay, so from this modifying organism, you are getting the desired proteins. Desired proteins. So now extracting these desired proteins in a sterile condition, we call it as bioprocess engineering. So whatever the proteins that you are getting, that you are filtering out, extracting it out, and making it in a sterile condition, packaging and producing the producing the desired product, that you call it as bioprocess engineering. It's all about the technique that how you will extract a protein or how you will extract a particular product from the organism in a sterile condition. So that's why it is called as the process. As you are processing here, processing the product into a pure form without having any contamination of the bacteria or any other things. Okay, so remember bioprocess engineering, it involves the maintenance of a sterile condition and extracting the pure product. We call it as bioprocess engineering. So write down this definition. Okay, so this is the important definition. So write down. So according to EFB, that is, EFB means European Federation of Biotechnology. Okay, so what EFB has given the definition for biotechnology, it is the integration of natural sciences and organism cells, parts thereof, and molecular analogs for products and services. Guys, understood what's the meaning of this definition?
No, sir. I didn't understand.
Okay, so biotechnology, it is nothing but integration of the natural sciences. What are natural sciences? That means your bio, physics, chemistry, engineering. So all these comes under the natural sciences. Okay, and you are taking the organism, you are taking the cell, you are taking the parts of the organism or parts of the cells and making a molecular analogs. That means you are creating or modifying molecules. So you are creating or modifying the natural molecules. Okay, so those molecules will be mimicking. So what is meaning of analog? That means you are creating a molecule which is also same like, okay, like a natural one. So you are making a clone or a copy. So mimicking natural biological molecule. So what I'm saying is, first suppose let me say our insulin is produced in our body from the beta cells of pancreas. I am making this insulin. What is this insulin? It's nothing but a molecule. I am synthesizing this molecule in other organism. Let's say E. coli. How I'm doing? I'm mimicking this insulin by introducing the gene into the other bacteria. Okay, so that this E. coli can produce the insulin same like the insulin that is produced in our body. So what I'm doing, I'm mimicking the natural biological molecule. So that's what meaning the molecular analogs. So that they are created the product and services. That means you are creating different products. It can be the medical product, or it can be agriculture product. It can be the industrial product that can be used in the industries, environmental product. So let's see how does the medical. First, suppose you're making the vaccines, or you're making certain drugs, therapeutics. Okay, or you are doing you are using certain molecules that can also act like a diagnostic tools. In the agriculture, you are making the crops that are modified genetically, like the genetically modified crops. You are using pesticides that's made from the organism itself. Those are called as the biopesticides or biofertilizers. Industrially, you can create like cosmetics, okay, biofuels. It can be even the enzymes. So all those FND, food and develop drugs, you can synthesize all those. Environmentally, you can use certain products that acts like cleaning the pollutants. For suppose like bioremediation, we are using certain bacteria, viruses, those will clean the environment. So you are modifying all these organisms to get these products and services. So that means you are using the natural sciences like biology, chemistry, physics, engineering, all these, so that you on the organisms or cells, so that you are creating these products by mimicking the original products that are naturally produced. Okay. So remember, biotechnology is the combination of all these natural sciences with the living organisms. Okay, in order to create the product and services that will improve the medically, agriculturally, industrially, and environmentally. Understood? So what's the definition given by the EFB? Your molecular analog, you can write it like creating or modifying molecules to mimic natural molecules or biological molecules that are used for the drug development, for creation of the drugs, diagnostics, or you can say that can be used for all these things.
So all of the above.
All of the above. So read it very carefully.
So, genetic engineering, right?
Genetic engineering.
Yes, sir.
Yeah.
Genetic engineering.
So what's the question? It's the technique of altering genetic material to change the genotype and phenotype of organism. Okay. So what is bioprocess engineering? What is bioprocess engineering? Just now I told you, guys. What is
So like
bioprocessing?
maintaining like a sterile condition and then
Yeah. So it's a maintenance of sterile conditions to extract the pure product. So does it involve in the altering the genetic material? No, it's only just the process for extracting the sterile condition. And what is selective breeding? Come on, tell me, what is selective breeding?
So you pick the organisms with the desirable traits.
Yeah.
And then you.
Yeah. And what is genetic engineering? It's the technique where you will be altering the genome of the organism so that you will be getting desirable genotype and phenotype of the organism. Okay. So the answer is 3. What about the others? Why the others are not opening their mouth? Okay, guys, just only 3 slides left. Okay, recombinant DNA technology. So whatever that I'm saying, I'm altering the genome of the organism. That's called as rDNA technology. You can also call it as recombinant DNA technology, which was first introduced by Stanley and Herbert in the year 1972. So what do? We will be involving the gene cloning and gene transfer. Remember, in this technology, we are doing mainly 2 things. One is gene cloning and gene transferring. I'll explain you in detail what is gene cloning and gene transferring when we talk about this rDNA technology in detail. Okay. So it's nothing but introducing the desired DNA sequence in the organism. So as I said earlier, whatever the insulin genes are there in the humans, we are introducing that genes into the E. coli. So the E. coli is now started producing the insulin. So that we do by rDNA technology. So how many of you remember the Hershey and Chase experiment?
So it was like E. coli. They had used like
Yeah.
radioactive phosphorus and radioactive sulfur.
So what happened there?
So like they had firstly like dipped the E. coli, sorry, a bacteriophage in radioactive sulfur, which basically makes the proteins radioactive in the bacteriophage, and the another ones in phosphorus, like radioactive phosphorus, which made the genetic material DNA radioactive, and then they had like induced those bacteriophages. So the bacteriophages then injected like the
Yes,
genetic
yes.
material like that. Yeah.
So there is a bacteriophage. So what are bacteriophage? Is that a virus or bacteria?
Virus.
Okay. So virus, that virus, what does it do? So bacteriophage, these are the virus that will infect the bacteria. So what does this bacteriophage will do?
So it transfers its genetic material into the E. coli.
Yes. So what does it do? It will transfer, it will not send the protein, but it will send the DNA into the bacteria. And we know there is a bacterial DNA. So whatever the DNA that it sent here, that will become part of this DNA. That will become part of the bacterial DNA. So this is bacterial DNA and this is viral DNA. So this viral DNA will be introduced into the bacterial DNA. So you say this is the bacterial DNA and this small part will be the viral DNA. So what happened? Now this bacteria will start doing translation and produce the proteins. So whenever it produces the bacterial proteins and also viral proteins. So here the viral proteins is nothing but it is producing the virus. So what actually happened here? Some other organism DNA was introduced into some other organism DNA. Now this organism got modified and it is producing those added DNA, those added proteins as well. That means this is, let's say this is A DNA, this is B DNA. So I introduced A DNA into the B DNA. Now B DNA got modified into B dash. Now this B dash DNA is producing the proteins that are related to A as well as B. That means, can I say this organism is genetically modified? Here, whatever the E. coli was introduced by the bacteriophage DNA, is it now E. coli is genetically modified?
Yes.
Guys, you understood what I'm saying here? The others.
You're introducing new DNA, right?
Yeah. So after introducing the DNA, now it's producing some there's another type of proteins which it is not produced initially. Mandara, Tanzila, Shirley,
Yes, sir.
you guys understood what I'm saying here?
Can you repeat that, sir?
Okay. So listen carefully. So what happens here is in the Hershey chase experiment, there is a bacteria. The bacteria is having its own double strand circular DNA. So let's say this is the bacteria, which is E. coli. And there is a virus that's called as bacteriophage. So it is a virus. So what is virus? Virus is nothing but a protein coat. Virus is nothing but a protein coat and the genetic material DNA. Okay. So what happens is this virus will do transduction. That means it will introduce its DNA into the bacteria. So whatever this DNA is there, it will be introduced into the bacteria. So after introducing the, let's say this is viral DNA, vDNA. This is bacteria DNA. So this viral DNA now will become part of the bacteria DNA. That process we call it as, what do we call it as? Come on guys, tell me, what do we call it as? We have studied in molecular basis of inheritance.
Sir, can you please repeat?
Here the bacteria DNA and there's a viral DNA. This viral DNA now will become part of the bacteria DNA. That process, what do we call? Or tell me, the transferring of the genetic material into the bacteria, we call it as
Transduction.
transduction. Okay.
Yeah.
So here the transduction will happen. So this viral DNA and bacterial DNA, okay, is created. That means whatever the viral DNA that is introduced into the bacteria cell, that viral DNA will collab with the bacterial DNA and it becomes part of the bacteria DNA. So this one, we call it as recombinant DNA, let's say. Okay, that means it is genetically changed. That means initially it is only the bacteria DNA, but there is a new set of DNA is also introduced. So when this DNA will undergo translation process, it will create the bacteria proteins and also the viral proteins. Yes or no?
Yes, sir.
So that same technique we will be using in our DNA technology. That means we will introduce the foreign DNA into particular organism DNA. Now that organism is genetically modified organism that will produce the new DNA, whatever that we introduce, those proteins as well. As this E. coli started producing the bacteriophage proteins, here, whatever the genetically modified organism, it also produces those desired proteins. Understood what we are doing in our DNA technology? It's okay. We'll talk about this in detail. Just now you understood what is the basic mechanism, basic funda behind this genetically modified organisms, how we are doing our DNA technology.
Yes, sir.
Okay. So you can see here. So let's take, there is a plasmid with antibiotic resistance. So you can see here. So actually there's a plasmid. What is plasmid? What is plasmid? Extra non-chromosomal
DNA.
DNA that present in the bacteria. We call it as plasmid. So these plasmids in most of the bacteria, they contain antibiotic resistance genes. Okay. So remember, they contain antibiotic resistance genes. That means whenever you give a particular antibiotic to these organisms, those bacteria will not be killed. They will show resistance towards the antibiotics. For suppose you give tetracycline, the bacteria will resist towards the tetracycline. You give penicillins, it will show resistance towards those penicillins. You give ampicillin, it will show resistance to those drugs. So remember, there is a kind of plasmid that shows antibiotic resistance. So that antibiotics resistance is got by this DNA, whatever that looks in the orange. So this is our DNA. So I am cutting out with the restriction enzyme. That's a restriction endonuclease, which are molecular scissors. So they will cut down this DNA. So remember, this part of the DNA is antibiotic resistance gene. And I am taking another plasmid, let's say Salmonella plasmid. I'm also cutting exactly at the same point where the ends of ear will match. Okay. So do you remember the palindromic sequence that we discussed in molecular base of inheritance?
Is it when all the nucleotides are the same from both ways?
Yeah, we call them as sticky ends. Yes or no? We call them as sticky ends. Palindromic sequence. They will be having complementary nucleotides, so their ends will stick if we bring them near. It's like a magnitude of north and south pole. You bring them near, they will stick. The same way here, this end and these ends will be having the palindromic sequence. So whenever you bring near to this blue one, they will attach. If not it attached, you also use certain other enzyme called ligase, which acts like a glue, gum, that will attach this resistance gene with the Salmonella plasmid. And you will introduce this into the host organism. So when you introduce this into the host organism, so when this E. coli start making the proteins, they also make this antibiotic resistance proteins. So that's how you are modifying this E. coli. We'll study in detail. This is just a basic how we do our DNA. Is that clear?
Yes, sir.
Yes, sir.
So it involves in 3 steps. Step number 1, identification of DNA with the desirable genes. That means, for suppose in particular organism, I have a particular protein that is very, very good. So I want that protein. That means this is my desired protein. And this desired protein will be having desired genes. So we need to identify that DNA, which is coding for this desired protein. So that's the step number 1. So you need to identify the DNA that is coding the desired genes. So what do we do next? We will extract this desired DNA and we will introduce into some other host organism. So now this host organism will become genetically modified organism. So once we do that, we will be doing bioprocessing. That means you will maintain the introduced DNA into the host organism, and that host organism will be giving the offsprings. Those offsprings, after several generations, they will start producing that particular gene as the protein product. So now I'll take that product and I'll bioprocess it and I'll extract that in a pure form as a pure product. So this is how you will be doing the RDNA technology in a nutshell. We'll be talking each and every step in detail in the next class. You understood?
Yes, sir.
Yes, sir.
Is that clear for everyone?
Yes, sir.
Yes, sir.
Any doubts?
No, sir.